All right, so I'll just go ahead and jump in. So again, thank you very much to the organizers for allowing me to speak today. So the purpose of this slide is really just to orientate everybody here because you might not be thinking about circadian rhythms. Certainly I didn't 20 years ago. And so the purpose of this slide really is just to remind everybody that every aspect of biology and physiology is circadian regulated. Whether you're interested in cognitive function, gastrointestinal issues, if you're interested in neurohumoral factors, metabolism, something we're really interested in, as well as what we're going to talk about today, cardiovascular function. All of these things change over the course of the day. So here's just an example of some data on the left-hand side of the slide from a healthy volunteer. You can see this healthy volunteer right here when they go to sleep. There we go. When they go to sleep, both their blood pressure and also heart rate is going to decrease. And then upon awakening, you're going to get this steep rise happening. And as you can see on the right-hand side of the slide, when you average that, you take the average of multiple individuals, you can see that in addition to this steep rise in the early hours of the morning in blood pressure and heart rate, you also get this secondary peak and in the early hours of the evening, and that's something I'm going to come back to in a few moments' time. However, unfortunately, associated with that steep rise in blood pressure and heart rate in the early hours of the morning, you also get an increased incidence of MIs as well as sudden cardiac death. Now, that was first observed back in the 1980s by Mueller, and so what Mueller did was he lined up the usual suspects. He said, okay, so let's look at things like blood pressure, heart rates. Let's look at adrenergic stimulation. Let's look at clotting factors. Let's see what they're doing at that time of the day. And not surprisingly, he found that all of them were elevated in the early hours of the morning. So based on those simple observations, he came up with this model on the right-hand side of the slide, which is basically saying in an individual that has a vulnerable atherosclerotic plaque, that rise in blood pressure in the early hours of the morning is going to create a shear stress that is going to increase the likelihood that that plaque ruptures. That in alignment now with an increase in your clotting factors means that you're more likely to develop an occlusive thrombus and therefore have an MI and or sudden cardiac death. And really this model probably holds to this day. And this was developed back in the 1980s. So you might be wondering, well, why am I here? Why am I talking about this? Well, it's because since then we've realized that in addition to these extrinsic factors, so Mueller was interested in sleep-wake cycles, for example, and behaviors, and then all these factors that are outside the cardiovascular system, we now know that in every cell in the body are these cell-autonomous circadian clocks, these little molecular clocks that allow each cell in the body to know what time of day it is. So these are really transcriptionally based mechanisms, at the heart of which are two transcription factors that I'm going to come back to over and over again, called clock and BMAL1. When these two transcription factors heterodimerize, they bind to E-boxes and target genes, leading to the induction of those genes. Now, some of those genes are going to encode for proteins that make up negative feedback loops, such as those encoding for period and cryptochrome, as well as those encoding for the reverbs, reverb alpha and also reverb beta. And so when these proteins accumulate, what they do is they feed back on that clock, BMAL1 heterodimer, and they shut it down, such that you get these activity rhythms in BMAL1 and clock that last about 24 hours. So you'll have a 12-hour period where you're increasing activity, and then a 12-hour period where the activity is troughing. It's going back down. Now, all of these factors that make up the clock are all transcriptional modulators. So in turn, what they do is they change the expression of thousands of genes inside each cell in the body. That leads to thousands of proteins being altered over the course of the day, leading to changes in physiology. Now, what Steve Shea and Frank Shearer in Harvard did several years ago was they asked the simple question. These factors that Mueller thought were responsible for increased MIs in the early hours of the morning, are they being driven simply by our behaviors, or are they being driven by these endogenous clocks? Well, how do you do that in humans? Because that's what they wanted to do. What they did was they tried to dissociate the environment from these internal clocks by forcing people to live 28-hour days. So basically almost equivalent to shipping them off to another planet. So what they did was they forced these individuals to live in a light dark cycle that was 28 hours in periodicity, They forced them to eat and to sleep and to have physical activity with a 28-hour periodicity. Then they looked for factors, these factors on this slide, that still had 24-hour rhythms. Because if they still have 24-hour rhythms, they can't be being driven by behaviors. And so you can see here that in the equivalent of the early hours of the morning, you still get a rise in platelet activation and Pi-1 levels, even though these individuals are living a 28-hour day. And in contrast, the rise in blood pressure that you see in the early hours of the morning, it goes away. So that has to be being driven by behaviors. But instead, that secondary peak that I mentioned I would come back to, that still peaks with a 24-hour periodicity in the equivalent of the early hours of the evening. Now we now know that the clock BML1 heterodimer directly regulates Pi-1. So this is through luciferase reporter assays, and this was in human cells, something that Joe was mentioning, using human cells in culture. If you overexpress clock and BML1 and you perform a luciferase reporter assay, you can see that these transcription factors are directly regulating Pi-1. So what we can do now is we can now go back to that original model of Mueller's and we can modify it slightly. And we can say that the early rise in blood pressure creating the shear stress is definitely being driven by behaviors. But however, that increase in platelet aggregability is being driven by your endogenous clocks, which means you have to have a synchrony between your behaviors and your endogenous clocks in order to have an adverse event such as a myocardial infarction. Now, when these data first came out, I couldn't help but have a little bit of fun. I wrote an editorial here saying that you're basically going to have pie for breakfast whether you like it or not, because at breakfast time your Pi-1 levels are going to go up and you cannot control that. Well, what happens when things go wrong? So what happens if you cause some kind of circadian dyssynchrony? And in humans, the easiest way to do this is through shift work, or if you're a resident in the hospital or a nurse, you're going to be doing this all the time. And unfortunately, we know that shift work is associated with a whole host of diseases, including increased risk of cardiovascular disease. And here's just a meta-analysis showing that there's about a 25% increased risk of adverse ischemic events seen in shift workers. Okay, so now we're going to move away from the onset of MIs, and we're now going to talk about what we're studying in our lab, and that is tolerance to an MI, so ischemia reperfusion tolerance. In humans, there is some data out there, and I would manage to find 5 different studies looking in humans at the time of day at which there seems to be greatest damage to the myocardium following an ischemic event. Turns out that it's also in the morning. So unfortunately, in addition to having an increased risk of having an MI in the morning, you also have increased damage to the myocardium if you have an MI in the morning. So it's a double hit, I'm sorry to say. Now, what several studies have done is they've recapitulated those observations in humans, and they've seen this also in rodent models. And again, the majority of these studies find that at the beginning of the active period is when you see the greatest amount of damage to the myocardium in response to an ischemic event. There's a little bit of variability. A couple of studies were outside that range, but the majority of those studies are inside this range right here. And again, if you perform some kind of intervention, unfortunately we don't have data in shift workers about tolerance to an MI, but if you take animal models and you disrupt their circadian environment, or you disrupt the internal clocks by, for example, genetically targeting that clock gene, you find that there is worse ischemic tolerance. Excuse me, I'm losing my voice here. So the data on the left-hand side of the slide, is a study in which Tammy Martino induced a myocardial infarction in mice and then disrupted the light dart cycle and found that there was greater infarct expansion in these mice here with a disrupted circadian environment. On the left-hand side, on the right-hand side of the slide is a study again from Canada. This is Lori Kirschenbaum's group, where they found that in the clock mutant animals, there is greater damage to the myocardium following IR. Okay, so you might be wondering, what is the actual mechanism? How is this happening? What What is driving these daily rhythms in ischemic tolerance of the myocardium? Well, there's so many different parameters that we know will impact MI tolerance. And to be honest, here's just a review article. All of these things are circadian regulated. I can tell you that. And that's really where my lab comes in. So what we've been doing is we've been trying to identify what these circadian clocks are doing in different cell types. And the majority of our work has been in cardiomyocytes. And over the years, what we have found is that the clock in cardiomyocytes is regulating a whole host of processes ranging from electrophysiology, metabolism, and cellular signaling that ultimately changes the function of the myocardium over the course of the day. And more recently, we've been studying this axis right here, some of which you've already seen. So I told you previously that that clock BMI1 heterodimer induces reverb alpha and reverb beta. What I haven't told you yet is that in turn, reverb alpha and reverb beta can repress this transcription factor E4BP4. And we just published a paper in JACC a couple of months ago, really focusing on the role of E4BP4 in the heart. A few years ago, we studied these different clock components in response to an MI. So what we did was we mimicked what happens in humans by we induced an ischemic event at the beginning of the active period, and this was in rats, if I remember correctly. And then we looked at the effect on these different genes within the border zone versus the non-ischemic region. And the border zone is seen in black here. And you can see within 24 hours, you're already seeing differences in the expression of these clock components within the border zone, the region that had some ischemia. And in particular, you can see that E4BP4 has this twofold induction within 5 hours after an MI. It's quite dramatic. So this led to the question, is this axis regulating ischemia reperfusion tolerance in the heart? Well, we had some evidence in the past from some of our past studies. This was the first study. This is from my group. This is the first study looking in rodents if there was a time of day dependent oscillation in IR tolerance, and in fact there was. And what we found was that if you genetically ablate the clock gene right here, it leads to cardioprotection. You absolutely prevent that time of day dependent oscillation in IR tolerance, and it was protective. And that to us was quite surprising. Fast forward 8 years later, and Bart Stahl's group, what they did was they created a reverb alpha knockout animal, and using an ex vivo system, found that the reverb knockouts also have improved tolerance to an ischemic event. So most recently what we've done, and this is in collaboration with Min Z at UAB, we've taken our E4BP4 knockouts, and this is cardiomyocyte specific. We've taken these E4BP4 knockouts and we've performed ischemia reperfusion on them. And we find that they have worse outcomes. So knocking out E4BP4 is detrimental, which fits with this axis because when reverb alpha and beta or clock are knocked out, E4BP4 is usually chronically elevated and we see cardioprotection. If we knock out E4BP4, then it leads to worse outcomes. Now, if you've been paying attention, which I'm sure you have been, you might see that there's a little bit of discrepancy going on here. So I showed you the data from our lab was finding that if you knock out, if you knock out clock only in cardiomyocytes, it's protective. But however, if you disrupt the clock in the whole body, it's detrimental. That said to us that there must be other cell types involved in these rhythms in ischemia reperfusion tolerance outside of the cardiomyocyte. And the cell types that we wanted to look at were immune cells. And in fact, there is already evidence for neutrophils that they do play a role in these daily rhythms in ischemic tolerance. And this was a study, not from our group, but this was from Schloss, in which, first of all, you can see here that just in normal animals, there's this daily rhythm in neutrophil levels in the circulation, which are peaking right at the beginning of the awake period, right when we say that there's worse IR tolerance. Then if you induce an ischemic event at that time in the day, so at the beginning of the active period, you get this persistent elevation of neutrophils within the myocardium, which is associated with worse outcomes. So if you were to attenuate these neutrophils within the myocardium, you find that you also attenuate these day-night differences in ischemia-reperfusion tolerance. So this led us, in collaboration with Sumanth Prabhu, to start looking at a different cell type. We became very interested in monocytes because monocytes are also very important for ischemia-reperfusion tolerance. And in fact, what I'm going to show you here is MIs rather than ischemia-reperfusion. The first thing we did was we looked at animals that had heart failure induced by an MI, and we looked at the status of the clock within Ly6C high CCR2 positive cells. And we found that the clock was dysfunctional, that BMAL1 was chronically low, as well as the reverb alpha was chronically low. So we said, okay, well, why don't we do this? Why don't we just knock out BMAL1 in monocytes and see what happens? And so that's what we did. You can see here, when you knock out BMAL1 in monocytes, you get low levels of reverb alpha, and you get chronically high levels of E4 BP4. When we take those animals and we induce an MI in those animals, we find that there are worse outcomes. These animals have a pro-inflammatory status, and they develop fibrosis to a greater extent, and they also progress to heart failure at a faster rate. So we then ask the question, okay, well, if knocking out BMAL1 is detrimental, we hypothesize that knocking out E4 BP4 would be beneficial. And so that's exactly what we did. We knocked out E4 BP4 in monocytes, and when we did that, we found that it had an anti-inflammatory effect during MI, and it also, those animals had better functional outcomes following an MI. Excuse me, I'm losing my voice today. Don't know why. Well, of course, we're not at the stage where we can genetically ablate something, so we have to rely on pharmacology in patients. And so we said, what can we do? Well, there are no drugs yet for E4 BP4. Okay, so that's one of the problems, and that's something we would really like to do. We would like to develop a drug, an antagonist against E4 BP4. But in the meantime, what we've done is we've utilized this compound here, SR9009, which will activate reverb alpha and reverb beta. And in doing so, when you activate reverb alpha and reverb beta, you will repress E4 BP4. And when we do that, and we take animals that already have heart failure after 2 weeks after an MI, and we then treat them for 2 weeks with the SR9009, we find that we can reduce inflammation and we can attenuate the progression of heart failure even further. Okay, so hopefully I've convinced you that there are these so-called circadian clocks in every cell in the body, that these clocks do play a role in the onset of myocardial infarctions. Specifically, they are increasing Pi1 levels in the early hours of the morning. They are also influencing in a very complicated way the tolerance of the myocardium to ischemic events, and it's probably in a cell type specific manner. That if you disrupt these circadian clocks, it's detrimental. It increases MI incidence, and it also worsens MI outcomes. Again, we come back to this cell specificity issue where E4 BP4, for example, does very different things in cardiomyocytes versus monocytes in terms of ischemic tolerance. But really, we're very hopeful that we can utilize this basic science knowledge that we've been gaining perhaps for the treatment of patients in the future. Okay, and with that, I'd just like to thank all the individuals who have helped with these studies over the years, including folks at UAB and my various collaborators around the world, including Sumanth Prabhu, collaboration with the monocyte work. And with that, I'd like to thank you very much, and I'll stop there.